Professor Jiawei at UIEC, and his talk title is ClaimMiner Query Guided Claim Mining in Biomedical Literature.
Okay, hi, good afternoon. So, like Zhiyong just introduced, I'm at UIEC. I actually at computer science, but we do have a group of students working on biotext mining. Here, I basically just report what we have been working on. So if you look at this title, ClaimMiner, what is ClaimMiner? ClaimMiner means you go down to PubMed or any other research corpus. You write down certain claims you want to find and see whether we can find those claims out from the text. Okay. So I better first give you an example. Okay. If you look at the table actually is one. If we give you a query, say cisplan acute renal failure, one, like you get a certain drugs or proteins or chemicals. The other one is say certain diseases. We want to find whether you can find the claims in the PubMed. You can find those things actually closely related, like a claim. What it look like a claim? You look at the first three sentences. Actually, these are claims. The last sentence, it does contain cisplantin and acute renal failure, but it's just experiments, just a statement. So you really want to find claims, not just some kind of a statement contain those drugs and certain disease. Okay. So what do we claim? Context dependent versus context independent. Context dependent means you give me a query. You say what kind of thing you want to find, what kind of claims you want to find. Context independent, just say finding all the claims, but you can find millions of them. It's not so useful. Okay, what you really want to find is give me certain, say, drugs and disease or something. You can find whether it contains this. Of course, we do not say you will find it's positively related or negatively related. That will be the future step. Okay. So if you look at the related work, in the past, the majority of such work are on the supervised model. That means you give me a lot of annotated data. That's exactly the previous researcher presenter just say, you painfully just annotate that data. We don't want anybody to annotate anything, okay? So that's the one we call supervised one, may not be very good. So there are distantly supervised or unsupervised one. However, even these one, they based on certain online database reviews and semi-structured. They never happen in biomedical domain. What they got is, for example, some people develop a fake news detector or something. Okay, so there's some competition on certain kind of claims or something, but actually it's not really in biomedical domain, okay? So we work out this claim mining contain two major parts. One part is offline processing, the other one is online processing. Actually, offline processing takes a long time, and in the meantime takes lots of research effort. What we are doing exactly like today, the first keynote speech, talking about named entity recognition, relation extraction. We are doing this, okay? The only thing different is we actually not really doing annotation. We rely on large available database or like a dictionary or something. Later I'll give you a little detail. Then we do that. Whether we involve any neural network, we do involve a little deep learning stuff, but I'm not going to have time to discuss this. But then the second one is online one. The online one is taking the offline, work out these different entities and types and try to see whether it contains claims and how to rank claims. Let's look at it one by one. The first one is offline one. Number one is named entity recognition. What is named entity recognition? Basically, you give me a sentence, there are millions of them. You try to find whether this one is a chemical, whether this one is a biological process or genes or disease. You want to automatically identify this. To identify this, actually this morning, Sophia mentioned a very important thing is nested named entity recognition, because so-called nested is you look at this one is a organ, this one is the disease, this one chemical, getting together maybe just one disease, okay? So actually we developed method, we did not give you the detail because we don't have time, but we do the nested name entity recognition in very good quality. It is published last year. And with this, we further do this is we integrate with our Outwhole ER, which is based on phrase mining and deep learning on both direction, bi-direction RSTM. But we use our pattern-based integrate with this, get one called PAT-NER. If you look at the PAT-NER, you look at the results comparing with all the previous one with, some kind of Markov model or something. If you look at the previous one, you will find that this one actually gives you the very best result, okay? And for this best result, we actually have a demo and poster in this session. Actually, this co-author, Professor Mikkel, is there. We already put a poster there in the H session. Later, you're welcome to get it. So the interesting thing is, for this one, we can recognize 17 fine-grained types, 5 coarse types, 17 fine-grained types, and including organism, including fully formed anatomical structures, chemicals, physiological functions, pathological functions, all these things. Of course, different from Wei Wang's talk, we could not find 17 colors because it's very hard for human. So what we did is we made the 5 major ones, we have the color. The other one is you point out there, they will give you the subtypes, okay, by writing instead of by coloring, because we do not know how to recognize those colors. But this one is our demo and poster. If you're interested, go to our demo. We can show you right online our online demo, okay? On the papers you can search. So that's the first part is on name entity recognition. The second part is on relation extraction. The traditional method still using very heavy, human annotation. That's what the state of the art, okay? What we did was using pattern-based open relation extraction, okay? The pattern-based, we actually developed a meta-pattern-based approach and a relational tuple extraction. One paper was published in last year, BIBM, is WWPI's wide window pattern-based information extraction. What is wide window? Wide window means in the middle you may have a subclause, you may have some, longer, some other subphrases. Our pattern mining try to, partially parsing this part, and then some phrase, even in the middle, there are some substantial portion, we still can link them together, okay? Once you link them together, you probably can see, for example, if you look at a chemical activated gene, you will find lots of aliases. And those aliases all, grouped together, so you find the same kind of relations, okay? And with this, you probably can see, we can extract many same kind of relation with all the different instantiation, okay? This one we have the demo included in as well. Now we get into claim mining, okay? To do the claim mining, actually the first thing is to generate candidates. How to generate candidates is user give us, for example, you give us cisplatin, acute renal failure. Actually, we'll get this entity, cisplatin, will be expanded, and acute renal failure also as a disease will be expanded. With this, you try to do the matching in different ways. We match all kinds of words and entities and patterns. Okay. However, we have different ranking methods. So finally, the claim you ranked, the most important claim, most confident claim will be ranked on the top. So we have 3 ranking criteria. One called exact query entity. That means you match exact query entity comparing to synonyms. The synonyms, acronyms, but not exact match will be ranked slightly lower than the exact one. The second one is a query entity versus query words. That means you take the whole entity instead part of the words to do mapping. The whole entity matching will be ranked higher. The third one is query match the meta pattern. Remember, we got this meta pattern? If you match the whole meta pattern, you'll be ranked higher than just match part of it. Okay. So with this, of course, there are detailed functional module, but I will not really have time to go over this different function module. But you know, these are 3 functions, related to all these 3 different, ranking criteria. Okay, these are 3 different functions. Finally, all these 3 things, we integrate them together. We get a general claim score. Okay, that means the most important one, most matching one will be ranked higher. The other one will be ranked lower. Okay, so I better show you some experimental results. You will see it could be quite interesting. The experimental results, one is we took PubMed raw corpus with 28,000 abstracts as our experiments. Okay, and with this we get into 30 relation pairs from the previous database. There's a comparative taxonomics database. We take that database. You actually can see the matching there. Interesting one is the left-hand side is matching we call general typed query matching. The right-hand side is concrete query matching. What's the difference? The general typed query means you actually, if you do not know what disease, for example, you say arsenic, okay, but you do not know what disease may cause. You just get a general one. You get a dollar disease. You match any kind of disease as long as it's in the disease type, okay. Then the second one is you can match concrete query. For example, you can say arsenic, then cancer, or skin cancer, or something. Then you will match only this claim contain cancer or skin cancer. So otherwise, you match any kind of disease. Okay. Then if you type this query, we are going to based on our named entity recognition and relation extraction, all these are indexed. So based on this, based on a ranking score, you are going to match them in the 3 combined criteria. Finally, you rank them together. Okay. So you probably can see these are the ranked claims. That means the top ranked one, you're pretty sure they are good matching good claims. Then you go down, it may not be very good claims, but sort of like this. So you probably can see we actually did the analytical performance study. What about you only use words? What about you use words plus entity? What about you use all our features, including patterns? You actually can see the NDCG, which is an information retrieval measure. The higher value, the better. You actually can see the ClaimMiner will get 0.965. That's a pretty high matching. Simply says the claim we retrieved are really good claims. Okay, so. And you probably can see we give you some certain experiments. For example, chemical-induced disease, disease induced by chemical, or these different kind of things. You actually can see what kind of sentences we can grab and what kind of a soft matching and hard matching, okay? So, of course, there are still lots of work to do. As far as we know, not so many people really want to dig the claims out, okay, which is not that easy, and especially without annotation, and we did it. Of course, lots of future work to be done. Okay, so what I will show you is the first thing is we have 3 ISMB posters and 2 demos. Of course, we don't really have demo booths. You go to our poster, you say which demo you want to look at it. We will show you because all these via internet. It seems to be internet access is quite good here, so we can show you on the internet. Okay. The posters, one is on, I could not see that clearly. One is on text mining for biological discovery. The other one is on GitHub. The GitHub, what we did was, I just give you a simple introduction, is we organized GitHub, the biomedical GitHub in multi-dimensional cube structure. So you say you want to find a certain biomedical GitHub concrete directory, concrete links. You say what you want, okay? In a multi-dimensional way, we were going to show you, where those software are. Okay, those are the GitHub repository we actually doing data mining and organize it. The last one is ClaimMiner. And for the demo, we want the set search. Actually, the last year, with Zhiyong Lu, we actually published a demo in the KDD conference, was attract lots of cloud. Essentially, is you try to use many different entities and like a biological, like you give 10 genes, one disease. You say, what are the biomedical literature contain most of these genes and disease? You are going to find it. Otherwise, the current, you go to PubMed, you may find only 1 or 2 genes because they are so popular. They basically, the other genes, you may not be able to find it. Okay, so this is set search. But anyway, I think that this is one demo. The other demo is, I just show you this text mining demo. You will be able to grab any article to see there are 17 different types. Sometimes it's nobody curated, okay? But we actually automatically did it based on the dictionary, okay? Final one, I just show you, there's like a Ping and Wei Wang just did one on the biocare project. To some extent, we are also contributing. If you look at the people working on this, besides Ping Wei Wang, there's Cassie Wu from University of Delaware, and me from Illinois. We work together, okay, with the previous BD2K. So we will be gradually contributing to this, biological entity discovery and mining, okay. So I welcome questions.
Thank you very much. Questions?
So does the claim has to be in one sentence?
Uh-huh.
So in the ClaimMiner?
Oh, ClaimMiner.
Is it one sentence?
Yes, that's a very good question. The current ClaimMiner, you get a claim within the same sentence. Definitely, this is a restriction. We actually are working on cross-sentence, but they are closer. Yeah. But the current is within one sentence. Yeah.
Any other questions? I have a question. So when you showed the evaluation, the NDCG was very high in the 90s. So
Yes.
I was just curious, what was the gold standard?
Oh, actually, the gold standard was more like, because there's nobody actually in the past that did this claim mining. So what we did was the students, they actually give all the marks, say this one's a claim, this one is not a claim, okay? So to that extent, there's no real gold standard. It's within our own research group. Other students mark them, then these students just test them. Yeah.
Any other questions? Well, thank you very much.
Okay, thank you.
So we'll now continue with a few short talks. The next one will be Dr. Sanfen Zhu from Fudan University in China, and his talk is a full mesh, improving large-scale mesh indexing with full text.